Hi Bio30s! Today we are talking about traits that are carried on the X or the Y chromosome. Remember that before we were talking about traits that were autosomal, which means that they were found on human chromosomes from pair 1 to 22, if I was looking at a karyotype. And this time we're talking about pair 23, which is either XX if you're a female or XY if you're a male. We're going to talk about how they're different from our normal autosomal traits and how they're carried a little bit different. They end up with different ratios in our offspring depending on if you're male or female. So just a few things to mention before we get started. So remembering that chromosomes carry genes, and that's where we inherit things from, is in our genes. Homologous chromosomes separate during meiosis. Gametes always carry half the number of chromosomes as a full somatic cell. And when we are lining up on the metaphase plate, chromosomes assort independently, which means that the maternal homologous chromosome and the paternal homologous chromosome not necessarily always on the same side, so we end up with different combinations in our sperm and our egg. So we talked all about our scientist called Gregor Mendel. He's the father of genetics. But today we're talking about the next scientist that did some really amazing things for genetics, Thomas Hunt Morgan. We just call him Morgan. He lived a little bit later than Gregor Mendel, and he mostly studied fruit flies. So he's from America. He worked with fruit flies, and he basically used these fruit flies to figure out that some genes are carried on your sex chromosomes. So based on your gender means that you have a higher or a lower chance of getting certain traits. So when I'm looking at fruit flies, the reason that you would pick fruit flies to study is because they live very, very short lifespans, only 10 to 15 days. They produce a whole bunch of eggs all at once. I can really easily make genetics experiments and I can repeat them over and over and over again. If I have the same type of parents, I can cross them multiple times and always end up with hopefully the same ratios of offsprings to prove a point. They mature really quickly, so they become sexually mature very, very quick, which means that they're able to reproduce and then I can use them for genetics very quickly versus other organisms. I can tell apart males and females. So if I look at this picture, if you look at the bottom part of the fly, it looks a little bit different, more striped in the female and it's longer in the female. The male has a little bit less stripes and it's usually darker right on the end and it's a little bit smaller in males. So for us to be able to tell them apart is very important and easier for us to work with. They're also really small. So if I was using something like a mouse, for example, you'd have to have like cages for mice, which are a little bit bulky. These guys, you can a whole bunch of them in a test tube, and you can put test tubes in racks, and you can have a whole bunch of test tubes, okay, kind of sitting on a counter. It's going to take up a lot less space. And there's a whole bunch of characteristics, just like Mendel looking at his peas, there was like shape or size or color or flower color or all of these different kind of characteristics. There's the same kind of idea with these flies, so they can have different eye colors, wing types, okay, whether they're round or kind of shriveled wings, where they're actually sitting, how many wings there are, the legs that they have. There's a whole bunch of different characteristics that Morgan could look at. And they have a small amount of chromosomes, which is easier for us to look at than looking at something like a human. So I'm going to be looking at eye color today. And there are red eyes or there are white eyes when I talk about fruit flies, and red is dominant over white. But this trait is found on the X chromosome. So instead of writing it like big R, little r, that's how we wrote autosomal traits, we're going to write it like it's connected to the X chromosome. So it's a superscript right beside the X as a big R equaling red and a little r equaling white, okay, connected to the X chromosome, which is going to show that it's actually on the X chromosome, and it's going to kind of play out in our Punnett square a little bit differently. So we're going to look at two different crosses to start out with. First, we're going to look at a homozygous dominant female who has red eyes, and we're going to cross it with a white-eyed male. Okay, remember males are X and Y, so they only get one X that's connected to an R, and they, if they're going to be white, would have to have a little r. So if I'm going to look at the gametes and put them into my Punnett square, okay, we have homozygous dominant, so big R in each gamete, And in our males, again, male has an X and a Y. The R is only connected to the X chromosome, so they only get one R. So in this first box, we would end up with a heterozygous female. We would also end up with another heterozygous female. Okay, we have a red male and another red male. So all of our offspring are red, but because when you're looking at these organisms and when you're looking at their letters, you have to separate the males and the females. Remember, we talked about males having a different kind of stripy pattern on the bottom part of them, and females looking a little bit different. So when I look at the actual fruit flies, I can tell them apart, and I would be able to tell if they have different eye colors. So your phenotype would be a 2-to-2 ratio because you have 2 red females and 2 red males. But remember, you're always taking it down to the smallest possible ratio. So you end up with a 1-to-1. And you would have the same for genotype because you have 2 heterozygous females. And males have different letters. They have X and a Y. So they would be different, separated in my ratio. Again, 2-to-2 being taken down to a 1-to-1 ratio. I'm going to take 2 individuals from my F1 generation and I'm going to cross them. So all of my females were heterozygous and all my males were just red. So I'm going to take 2 of those and see what I end up with. So I end up with heterozygous versus a red. I have a red female, homozygous dominant. I have a heterozygous red female. I have a red male, and then I have a white male. So this is the first white male that I end up with. And my phenotype is a 1-to-2-to-1 ratio because I have 1 red male, 2 red females, and 1 white male. But my genotype, because they're all different, I have a 1-to-1-to-1-to-1 ratio. So when Morgan was doing these things, normally looking at what Mendel got with his parental generation versus his offspring versus his F2 generation, he was getting certain things, and Morgan wasn't seeing the same things as he was, and he was seeing differences between males and females. So after doing this, he concluded that the genes that control eye color must be on the X chromosome, and that Y does not carry an eye color, and that's why males were ending up more likely to have white eyes because they only needed 1 little R to have a white eye, and that males could never be heterozygous. So they either are dominant or they're recessive. They couldn't be carriers of a trait. So sex-linked traits, this is the title of the slideshow, are almost always on the X chromosome. Remember, we talked about Y chromosomes being really, really small, so they're not able to carry a lot of genetic information besides telling males to produce testosterone. So the Y chromosome is only basically exclusively used for sex determination. Okay, at 8 weeks, the embryo decides if it's going to be a male or a female, is the presence of a Y chromosome telling them to do that. X-linked traits occur more in males than females. Y-linked traits occur in males exclusively. So why is that happening? Okay, first of all, males only get 1 X chromosome, so they only need 1 recessive allele to have a recessive trait, where you normally need 2 recessive alleles. So it's more common for them to get something that's recessive because they only actually need 1 letter instead of having 2. And then Y-linked traits only happen in males because females don't have a Y chromosome. So obviously they wouldn't be able to get a Y-linked trait. So we're going to do this problem together. Okay, this time we're going to take a red-eyed female and cross it with a white male, but the red-eyed female is going to be heterozygous. So again, remember, red is dominant over white and it's connected to the X chromosome, so our legend will look like this. Heterozygous female and a white-eyed male. This is what my gametes look like. I'm going to put them into my table, and I'm going to end up with a heterozygous female, a white female, a red male, and a white male. So my phenotype is a 1-to-1-to-1-to-1 ratio because they all look different. And my genotype would be the same because they all have different letters. So there are some other examples of traits in humans that we find that are connected to the X chromosome. So example number 1 is colorblindness. When you are differentiating between red and green, that gene is found on the X chromosome. So if you are normal vision, you would have a capital C. If you're colorblind, you would have to have 2 of these little C's if you're female, or 1 if you're male. So my phenotypes, my options for females, you can be homozygous dominant or heterozygous and have normal vision. To be colorblind, you have to have two little c's. For males, because you only get one X, there's only two options for genotypes or phenotypes. You're either normal or you're colorblind for phenotypes, and genotypes would be you're either a big C or you're a little c. So colorblindness is more common in males. It's harder for females to get it because they have to have two little letters. So they would have to have a father that is colorblind and a mother that is at least a carrier to be colorblind. So the second example is called hemophilia, which causes a person to have a hard time clot their blood. Okay, and basically what happens in this person is they're not able to build the clotting factor that causes fibrin to be created. If you guys remember this from Bio 20. Okay, if you don't, I actually have another video on my YouTube channel about clotting. If you're having a hard time remembering it, you can go quickly watch that video. So the first thing that happens normally when a blood vessel gets broken open because you're injured is platelets kind of start to form this temporary clot. And then what happens is you go through a series of processes to eventually create fibrin, which is like this meshy net that goes over top of the clot. So this person just basically isn't able to create that fibrin because they're missing one of the intermediate steps to create it. So because that happens, this person is not able to properly clot their blood, which means that they would not be able to stop from bleeding out. So that could potentially kill a person if they're not able to treat this. Okay, nowadays they can take a synthetic version of this clotting factor and they're good to go. But this information is found, for this gene, is found on the X chromosome. And we more often see it in boys than girls because the same reason for colorblindness, because females have to have two little h's to have hemophilia. Males only need one little h to have hemophilia. So you end up having, okay, sometimes lots of blood pooling in their joints or, again, bleeding out internally if they get a bruise, something like that. Okay, but again, nowadays it's actually fairly easy to manage this. You have a needle that you have to take every couple days that with this clotting factor in it, and they're usually pretty good to go. It's a pretty easy thing to manage nowadays. So some people that you guys might know with hemophilia in real life, okay, famous people. We have this actress right here, if you guys have ever seen the Lizzie McGuire movie, okay, she is the teacher in this movie. Queen Victoria and her son, okay, both had hemophilia. It's rumored that both Mother Teresa and also Abraham Lincoln had hemophilia. There is no scientific proof of this, but it's rumored that they had it. And then also, if you know any Russian history, the last Tsar of Russia, which was like the king, his son, who is the brother of Anastasia, if you guys have seen that cartoon movie that was made in the 90s with Anastasia, her little brother Alexei had hemophilia as well. So again, these people would have to be really careful about getting cuts and things like that because they are not able to clot their blood as well as regular blood clotting people. So this brings us to the end of my sex-linked trait video 1. I'm going to be making a second short video to explain a few more concepts. Watch the second video as soon as you're finished this one.